Hello, and welcome to the SimpleMed Respiratory Lecture Series. My name is Dr Madeleine Swannack, and I am the Respiratory Lead. Today's lecture will be on the development of the respiratory system. This lecture is aimed at preclinical or Phase 1 medical students and allied health professionals, and so it is pitched at that level. Our learning objectives today are to be aware of a basic outline of the embryological development of the respiratory system, and to understand two of the basic neonatal complications of embryological development, surfactant deficiency leading to acute respiratory distress syndrome, and tracheoesophageal fistulae. Before we start to look at the development of the respiratory system, it is important to divide the respiratory system so that we can understand the development of each portion. The respiratory system is divided into the conducting portion and the respiratory portion. The first is responsible for warming, moistening, and conducting air to the respiratory portion, which is the site of gas exchange. This means that the trachea, bronchi, primary and secondary bronchioles are part of the conducting portion, and the respiratory bronchioles, alveolar sacs and alveolar themselves, are part of the respiratory portion. The respiratory system begins from an outpouching of the primitive gut tube known as the respiratory diverticulum. This structure is derived from the endoderm of the primitive gut tube that will go on to develop into the gastrointestinal tract. The outpouching occurs from the foregut, which is the cranial end of the gut tube, and its outer surface is covered with mesoderm. As it is an outpouching, the respiratory diverticulum is continuous with the gut tube at the start of the development. To allow formation of two separate systems, a tracheoesophageal septum forms. By the end of week 4, the respiratory diverticulum splits into two bronchial buds. These will go on to form the conducting portion of the two lungs. Eventually, the respiratory portion of the lungs will form from the mesoderm that surrounds these bronchial buds. As you can see from the image, by the end of week 8, the 3 lobes of the right lung and the 2 lobes of the left lung have begun to form. It can sometimes be difficult to remember which lung has 2 lobes and which has 3. So one trick to remember this is that there are only 2 lines needed to write the letter L, so the left lung only has 2 lobes. A tracheoesophageal fistula is an abnormal connection between the epithelium of the trachea and the epithelium of the esophagus. Embryologically, this occurs because the respiratory diverticulum and the foregut are continuous, and if there is a defect in the formation of the tracheoesophageal septum, this connection persists. As you can see in the diagram, there are 5 tracheoesophageal fistulae depending on the location of the fistula and the continuity of the esophagus. Type C is the most common. A tracheoesophageal fistula often presents in an infant with feeding difficulties, breathing difficulties, or choking episodes. All types of fistula need surgical management to allow for feeding without aspiration and to ensure continuity of the esophagus. The period between 8 and 16 weeks gestation is known as the pseudoglandular stage. The conducting portion of the airway continues to develop, forming bronchioles from tertiary bronchi formed by the end of week 8. At this point, the mesoderm that will develop into the alveoli has not completed development, so there are no formed alveoli. This means that no gas exchange can take place in these lungs. The stage between 16 and 26 weeks is known as the Canalicular stage. Throughout this 10-week period, the respiratory bronchioles develop by budding off the bronchioles formed during the pseudoglandular stage. The respiratory bronchioles are the first structure to form in the respiratory portion. As the alveoli have still not developed, there is only very minimal potential gas exchange surface, and if born at this stage, the baby will be very unlikely to survive. Therefore, viability is only possible after the start of the next stage of lung development. This has implications for the threshold of viability, which in the UK is set at 24 weeks of gestation. The final stage of respiratory system development takes place from 26 weeks onwards and is known as the terminal sac stage. The alveoli begin to develop around 26 weeks gestation. Within the alveolar walls, there are two main cell types. Type 1 pneumocytes make up 90% of the alveolar wall, are made up of squamous epithelium. Type 2 pneumocytes make up around 10% of the alveolar wall, and are made up of simple columnar epithelium, which produce surfactant. Surfactant is made up of a mixture of phospholipids and proteins and plays a key role in reducing the surface tension within the lungs. Due to its hydrophilic and hydrophobic portions, it lines the surface of the fluid within the alveoli, reducing the surface tension. This means that the alveoli are able to expand at lower pressures, which is helpful on reinflation of the lung at the end of expiration. Acute respiratory distress syndrome is a life-threatening lung injury that allows fluid to leak into the lungs. In premature babies, ARDS is a complication of the immaturity of their lungs. It is related to the lack of surfactant production in immature lungs. Lack of surfactant means that when the alveoli collapse at the end of expiration, the pressure that the tiny lungs of these premature babies can exert to reinflate the alveoli is not enough to overcome the surface tension. Giving maternal corticosteroids when the mother is in preterm labour can help to mature the type 2 pneumocytes to encourage surfactant production, which helps avoid ARDS post-birth. That is the end of this lecture. Be sure to check out simplemed.co.uk for hundreds of free articles, thousands of free questions, and any support with questions that you might need. Thank you for listening. Goodbye.